Hello everyone, welcome to the ITLS Academy. Today we are going to discuss the topic environmental sources which affect the quality of the milk. Okay, we all know that the milk are produced by a lactating animal and this production are done by our environmental. Okay, and milk handling equipments are also there. So the lactating animal production environment and the milk handling equipment are responsible for the contamination or the microbial activity into a milk. Okay, and some environmental factors which can be considered are discussed in in today's lecture, and these are what they are: air, milk handler, and the water supply. So now in detail we are going to discuss one by one, that is your air, milk handler, and the water supply. The first one is your air. Okay, so we are not considering the air is a significant source for this microbial contamination, because it is come from a movement, and air transfer the soil dust particle which have a microbial contamination, and this will contaminate the milk product. Okay, when this milk are exposed to the soil and dust particle which are exposed by the air, which are transferred by the air, then it will expose the milk, and sorry, then it will contaminate the milk and spoil the milk. Okay, and the sources expose milk surfaces like quantity or small quantities of soil which fall into a milking bucket when a milkman milking the milk, and sometimes microorganisms are enter into a milk via air which entering the milking machine during the processes. Okay, so bacterial count of air in cow sheet or parlor seldom exceed 200 CFU per liter, is usually are much less. Okay, and the micrococci amount less than 50% of aerial microflora. This coroniform and bacillus spores and a small quantity of the streptococci and generis are also present and account for remaining the microflora. So calculation indicate that airborne bacteria account less than 5 CFU per ml of milk produced, and thus bacillus spore consists 1 CFU per ml. Now see what is written in a slide. Air is not considered a significant source for a microbial contamination in a raw milk. Okay, we are not considering our air is a significant source. Through its movement, air transfers soil and dust particle from a microbial laden source into any exposed milk surfaces. Okay, so this soil and dust particle are considered as a source for the microbial contamination. Microbial contamination. This air and soil, and this air and soil come in contact with the uh, milking bucket during a hand milking, or microbes entering into a milk via entering the milking machine during use. Bacterial count of air in cow sheet or parlor sediment exceed 200 CFU per liter. 200 CFU per liter, and usually are much less. Micrococci account for less than 50% of aerial microflora. Corniform bacillus spore and small quantities of streptococci and generis also may be present and account for the remaining microflora. Calculation indicate that airborne bacteria account for less than 5 CFU per ml of milk produced, and the bacillus spore consists less than 1 CFU per ml. The milk handler, the second source of the contamination is the milk handler. Okay, milk handler means that the milkman who are milking the milk, they will contribute in the microbial load in a raw milk, either by using the dirt particle from the odor, or it also increases the aerial contamination through the isolated the air environment. Okay, and the risk of the contamination from the milk handler are very rare with if the milk are done with the machine. Okay, but if the milkman are done, then there is a possibility that it will they will introduce the pathogenic microorganism into a milk, and in that's why in many countries there is prohibition of the use of the human for the milking, so that they will avoid the human pathogens, because human are carrier of the pathogens, and when they are collecting the milk, then there is a higher chance of the transfer of the pathogenic microorganism into a milk from a milk handler. Now see what is written in a slide. When cow are hand milk, it is possible that the milk handler can contribute to an increased microbial load in a raw milk by dislodging dirt particle from the udder, increasing aerial contamination through accelerated air movement and contacting the milk which infected the hand. Risk contamination of milk from the milk handler are much less with the machine milking, but because of the possibility of introducing the pathogen into a milk by the handlers, many countries prohibited certain human pathogen carrier from taking part in the collection of the milk. Okay, now the next one is your the water supply. Okay, see, water are also have a sources of the microbial contamination in a milk, because they contain a free pathogens, even a fecal contamination. There are lots of cases occur in a farm which obtain a water, and this water are untreated water supply, like it will come from a borehole, wells, lake and springes and river, and they will contaminate with the microorganism with the fecal origin. Okay, so some common example of fecal bacteria which are present in the water are coliform, fecal streptococci, and clostridia. Okay, and some other microorganism are also contribute in a milk contamination by a water supply, like your saprophytic, which are origin from a soil or vegetation, may be present, like your pseudomonas species, coliform, and other form of the GNRS, bacillus spores, corniform bacteria, and lactic acid bacteria. Okay, so there the amount of these contamination which are found in a water, they are widely affected the quality of the milk. Now see what is written in a slide. This means that the water supply must be from approved source, free of pathogens and fecal contamination. Okay, that means that the water which we are use in an industry, that it will come from a approved source, and it is free from pathogens and fecal contamination. In many cases, farms obtain water from untreated water supplies, boreholes, wells, lakes, springs, and rivers that may be contaminated with microorganisms from a fecal origin. Example of common fecal bacteria found in water are coliform, fecal streptococci, and Clostridia. In addition, a wide variety of saprophytic microorganisms originating from soil or vegetation may be present, including Pseudomonas species, coliform, and other GNRS, bacillus spores, corniform bacteria, and lactic acid bacteria. The amount of these contamination found in water will vary widely. In the United States, potable water source must be demonstrated through the microbial testing to be free of fecal associated coliform. Also, the elimination of potential cross connection between the potable water supply and contaminated water sources such as watering through is required by our PMO guidelines. Okay, now the next one is your the microflora of milking equipment and its effect on raw milk. When if we use a properly clean, disinfectant and drain milking equipment, then there is a less chances of the growth of the microorganism or transfer of the microorganism. Okay, this clean and disinfected milk contact surfaces like your milk can, bulk can also consider the major sources of the bacteria found in the milk. Okay, when it is collected from the odor. Okay, it also contribute the factors are failure which reverse a wound or damage equipment part and in addition to the cleanness of the wall assemblies. Now see what is written in a slide. When properly cleaned, disinfected and drained milking equipment can be eliminating as a source for microorganism in raw milk. However, inadequately clean and disinfected milk contact surfaces, including milk can and bulk tank, are considered to be the major sources of bacteria found in milk after its collection from the udder. Other contributing factor are failure to repress wound or damage equipment part and inattention to the cleanness of the wall assemblies. The simplest form of milking equipment is the bucket used during the hand milking. Okay, this will contribute in a contamination into a milk. Okay, because this milk during a hand handling, milkman are used milkman are used. Okay, and the bucket both will consider as a potential source for the microbial contamination. Okay, in the place of this hand milking, if we use a machine milking, then it will require more than one teat cup cluster which help the milk flow into a receptor or a pipeline which are possibly passes through a recorder jar. Okay, like in case of milk being collected in a bucket, the milk is pooled into either a milk can or a bulk tank. When a pipeline is used, the pipeline transport the milk into a receiver tank, from which is released or pumped into a milk can or a bulk tank. Ancillary equipment may be used a strainer or inline filter, cooler that may be open surfaces in can or plate heat exchanger and the milk flow indicator and meters. Now, cow normally milk twice a dairy and the milking machine must be clean after each milking. Okay, if it is not clean, then it will contaminate the milk by our microorganisms. Because of the complexity of this milking machine, some of its component, milk residue and associated bacteria may not be completely removed from the equipment and tend to accumulate over a time. Okay, because this milking machine are what they are complex. That's why some of its component and even some milk residue and associated bacteria are not completely removed when we wash the equipment. Okay, and they will accumulate over a time. Except in cold weather, bacteria remaining in the equipment can multiply between each milking and their number can increase faster than can be visually observed. Okay, except in very cold weather. Okay, like you're in normal summer weather, we all know that bacteria require a temperature. If it get a 37 degrees Celsius temperature, then in every 20 minutes it will be double. Okay, that's why the number of the bacteria will increases and it can be visually observed. Thus, visually inspection of the equipment provide little assurance that the equipment has been adequately clean and sanitized. In practices, the contribution of contaminated milking equipment to the microflora of the milk cannot be assessed definitely by a bacterial count on a milk produced because of the variability in number and type derived from a cow udder. The most widely used method for determining the extent of the bacteria contamination of milk count surface is by using a sterilized liquid rinse then subject to a rinse to an SPC. In the same way that milk is tested, the type of bacteria in the rinse then can be determined through a assessment of colonies isolated and their ultimate identification. Okay, so as we test the sample of the milk, similarly, what should we do? We test the equipment of the milk. Either it is done by a sterilized liquid rinse, and either it is done by a sterilized plate count. Okay, then the bacteria are isolated and ultimately identify same as it is. Okay, the exact relationship between the number of bacteria recover from the rinse and available to the milk during the milking is not done. However, from result of repetitive rinsing or from other assessment methods, the proportional recovered by rinsing is known to be at least 10% of the number which are available for a milk. The proportional will be higher where bacteria have multiplied on the moist surface as a biofilm or in milk residue water has collected in poorly drained milking machine. This is because the bacteria present in a biofilm residue are released readily from the surface. So this is what is the diagram of principal types of milking machine used in a United Kingdom. The first one is your vacuum pump machine. This is your bucket cow shield machine. This is your milking pipeline and cow shield parlor. Okay, here a vacuum pump and motor are there, then a regulator and then a interceptor are attached with it, then a regulator. Here a long milking tube, then it is your bucket, it is your plaster, it is your vacuum tub. Here the milking are done. Here you make vacuum gauge, air pipeline. This is your teacup claws. Okay, now this is your, in the second one, this is your delivery pipeline, sanitary tap, vacuum gauge, receiver, releaser, milk pump, long milk tuber, air position, long pulse tube. Okay, now the next one is your a method that appeared to eliminate the problem of relatively efficient between microbial remove via rinses versus microbial removal via milk is in the line sampling technique. Using this method, a direct assessment of bacteria contamination of the milk from the milking equipment and bulk tank can be made. It has been pointed out that milking equipment must be contaminated heavily to a markedly increase the bacterial count per million of the milk passing through it. For example, to increase the bacterial count 1000 per liter of milk by 1 bacterium per ml, it required 1 million bacteria. Thus, to increase the count by 10,000 per ml, it required 10 billion bacteria. A milking installation consisting of 4 or 5 milking stall and jar together with a pipeline and a bulk tank has a milk contact surfaces area of approximately around 10 meters square and therefore would need to be contribute 1 billion bacteria on average its surface clearly where milking equipment. So this is all about the factor responsible for the contamination in a milk. I hope you have understand all the things. If you have any query related to this topic, you can ask me in a comment box, or for a further detail, you can follow the ITLS Academy on Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and subscribe ITLS Academy on YouTube and get our app from a Google Play. You can also ask your query to the helpline number that are already mentioned in your slide, that is 7080-833-450, and visit our website, that is www.itlsacademy.com. Thank you for watching. Please like, share and subscribe.